so much, Yoni, and the organizers for inviting me. This is actually my first time in Israel. I've been trying to come since 2013, since the basal ganglia meeting, and then with Yoni through our BSF grant, so kept canceling, finally made it. I'll also say, growing up in Los Angeles, especially in my elementary school, I had a lot of friends and classmates that had ties to Israel. They would visit Israel. So when I kind of think about my development of my global perspective and different backgrounds, it was either India, where my dad's from, or Israel, where a lot of my classmates were visiting. So it's exciting to visit after many decades after this kind of early life vicarious exposure to Israel. So I'll be talking about, our lab does a lot of molecular profiling in specific cell subtypes in the context of addictive substances across the lifespan. I'll start with our adult work and then I'll shift into some of our developmental work at the end. So I decided not to put the US-centric graph up and just talk about the world. I mean, we had a lot of this yesterday about the background, especially with opioids, but here's just a worldwide drug activity, including opioids accounting for most drug-related mortalities in 2023. And as you can see, fentanyl or synthetic opioids are really what's actively out there. I don't think that works. And specifically fentanyl. I didn't put the US one up, but I did want to share this grant because if you want to know where the epicenter is for the most opioids, for the overdose deaths, especially due to fentanyl and other synthetic opioids, it's actually happening in the city that I live in Baltimore. You could see that the overdose deaths, this is from 2022, or like prior to 2022, 2018 to 2022, or really 2013 to 2022, where you have this huge rise in deaths in Baltimore, leading that compared to the national average in the US. And one thing that we've been doing to kind of tackle this problem in Baltimore is we started a new addiction institute that's really a holistic institute that the goal is to go from bench to bedside. So we have basic scientists, we have clinical researchers, clinicians, and then community engagement to really tackle this bigger problem of addiction. And I'm actually leading the efforts of the basic science aspect of this institute. So like many people here, my lab studies the reward and basal ganglia circuitry, most notably this ventral basal ganglia, which includes this nucleus accumbens area here. So I'll talk mainly about this area, but also some other areas are connected. I'll briefly touch on the ventral pallidum. So this is a illustration from my lab, so we actually didn't leave out the ventral pallidum. We had it in this illustration. But you can see that the nucleus accumbens is really poised in this areas where it's interconnected to all these glutamatergic areas, dopaminergic areas, and other areas, whether through inputs or outputs, that it's really playing this role in regulating natural rewards, but also behavior responsive to addictive substances or loss of reward, like anhedonia in the context of mental health disorders like depression. And again, my lab has mainly been focused on these two main neurons, these D1 or D2 expressing neurons. They're known as medium spiny neurons or spiny projection neurons. As Yoni mentioned, these D1 neurons are thought to, when you activate them, thought to promote reward. D2 neurons thought to kind of put a blunting on reward. We know that it can be a bit oversimplified. It could depend on the stimulus, the type of drug, or exposure. But I'll be talking a lot about some of the work we've found, some of the findings we found in D1 neurons in the context of opioids. So when my lab started to look at opioids, we were really interested in this whole addiction cycle where individuals are beginning to use drugs, they go through this binge intoxication, then they eventually stop and they have this, aversive withdrawal states. But then you get, with these protracted abstinence states, you get these negative affective states and these negative affective behaviors. And it's often these feelings of these negative affective states that can lead people to start to take drugs again, right? Because they're trying to self-medicate and relieve these negative affective states. And that goes back, that gets them back into this addiction cycle. And we were really interested in this because a classic study from Terry Robinson and Brian Kolb actually found if you give animals repeated morphine, then put them through prolonged abstinence, what they found is these medium spiny neurons, the ones with morphines, these neurons actually have this reduced dendritic branching. If you draw these concentric rings around them, do a shoal analysis, you see reduced intersections of the dendrites of these various rings and also reduced spines. This was done in all MSNs because we didn't have the tools back then to differentiate the different subtypes. But this was really interesting to us because we had had a series of studies in the context of stress. This is where we've used a social stress model, the social defeat stress model, where we have a experimental mouse exposed, we have this CD1 mouse living in this cage, and this experimental mouse is actually intruding on its home, so it can be quite aggressive. And after that 10 minutes of that aggressive encounter, the CD1 is housed on the other side of a perforated divider. And we do a number of behavioral readouts, and one is social interaction. What we find after, putting animals through 10 days of the social defeat stress, we get a group of animals that are, we call stress susceptible. So they're actually, if you put them in, look at their social interaction with a novel social target, animal they haven't seen before, you could see that the stress susceptible animals will run up but then actively avoid that novel target, whereas a control animal that goes to stress will interact quite a bit. And through a series of studies from Chase Francis, the first graduate student in the lab who just came back as faculty to University of Maryland, and Meg Fox, a former postdoc, what they found was that these D1 neurons actually have this reduced dendritic complexity. Along with that, they had reduced excitatory input and hyperexcitability, whereas the D2 MSNs had enhanced dendritic spines. So we thought this was really interesting because this is a state where we see these negative affective states, and we see these D1 neurons have this loss of dendritic complexity, which is similar to what these classical studies showed in the context of morphine abstinence, where you have negative affect. So Meg then wanted to look at these neurons after she gives animals repeated fentanyl. This is just 5 days in their home cage, in their drinking water. They only have fentanyl. There's either fentanyl or the water controls. And she first just wanted to make sure that this paradigm induces a response. So she did a naloxone precipitated withdrawal and found that the fentanyl group did have this higher global withdrawal score. And then through a series of studies with this fentanyl, but also through our stress studies, as I mentioned before, we see these negative affective outcomes. So here's an example with fentanyl where Meg actually just did a social preference test where you have a same-sex conspecific in one chamber here under a cup, and then you have an empty cup here. And she found with the water, animals that receive water, they're interacting quite a bit with the same-sex conspecific, shown here, but the animals that receive fentanyl, they're actually avoiding this novel same-sex conspecific. So they have this they have this reduced social interaction, suggestive of less motivation to interact, to have social interactions. And this is similar to what we see with our social stress. She also did other tests. One is the elevated plus maze, where you can look at an animal's time in this enclosed, safer arm versus this open arm. And you can see the water animals spend quite a bit of time exploring this open arm here, whereas our fentanyl animals are spending less time in the open arm and more time in the closed arm. And we've done a few other behaviors as well. So with this fentanyl absence, we're seeing these negative affective states, which we also see in our social stress models. So then Meg went in and looked at the morphology of these neurons just by using a combination of transgenic mouse lines and viral tools to sparsely label these neurons. And you can reconstruct these neurons and then just draw these concentric rings and look at their dendritic profile. What she found in the D1 neurons, if you look at water compared to fentanyl, you see this reduced dendritic complexity. If you do the shoal analysis, you see this reduced number of intersections at these various shoal radii. She also found reduced dendritic branches and dendritic length with fentanyl on board. And this was really cool because when we looked at our social stress conditions, we saw the exact same thing. These are also D1 neurons. You see this reduced shoal profile, reduced dendritic length, and reduced dendritic branching. So these very different exposures, fentanyl plus prolonged abstinence, or social stress, both conditions where you see these negative affective states, we see these D1 neurons losing their dendritic complexity. And one thing that was really nice is we've repeated this multiple times across various experiments, but we actually find that when we do a correlation of this area under the shoal profile and look at social interaction ratio, we always found that animals that have lower social interaction have this reduced D1-MSN dendritic complexity. We've done this in many different experiments. So I'm not going to go into all the details in the interest of time, but we've done a lot of work just dissecting out the molecular mechanisms, whether it's in stress in these D1 neurons or in our fentanyl abstinence. We've used approaches that allow us to isolate RNA specifically from these neuron subtypes. In the context of stress, we've found a transcription factor EGR3 that regulates the cytoskeleton molecule RhoA, that regulates other downstream effectors like ROC, and we've used various genetic tools or pharmacological tools to block these effects. In the context of fentanyl, we actually have done RNA sequencing on these D1 neurons as well as the D2 neurons. And the D1 neurons, we've done this big gene network analysis. And in one of these networks, we actually found a number of genes related to dendritic morphology or synaptic related genes. And we specifically found this transcription factor E2F1 was upstream a lot of these genes. So we've used a lot of tools to manipulate specifically these genes in D1 neurons, as well as pharmacological tools. I'll just show you a couple examples. This is in the context of stress, where we've actually blocked RhoA activity because we saw RhoA up. When you see high levels of RhoA, you often get reduced dendritic complexity. When we block that activity, we can restore the dendritic complexity. In the context of fentanyl plus abstinence, when this E2F1 was down, when we overexpress it, we can also restore that dendritic complexity. And then we've looked at a number of behavioral readouts. This is just one example: social interaction, stress or fentanyl or EYFP group. This is just a heat map where there's a novel social target here, and you can see that animals that went through stress or the fentanyl abstinence, they're spending often more times in the corner, not interacting here with a novel social target. Whereas when we block these molecular changes, restore dendritic complexity, you see more interaction with this novel social target. So we've really found these intrinsic mechanisms. But the next step is to see, are there extrinsic mechanisms that are causing this dendritic atrophy in these D1 neurons in stress or opioid abstinence? So one area we've been looking into is microglia, right? Because these dendrites are disappearing. It makes sense that microglia might be playing a role in this. And this is a project that's being led by Daniel Finker in the lab, but with the help from Ben Simpson when he was a lab to kind of set up this microglia analysis. So one thing that Ben did is he actually was able to reconstruct these D1 neurons and draw what's called this convex hole around it. It's like kind of wrapping a rubber band around this neuron and finding the shape that fits around the whole neuron. And what he could do when you draw this, you reconstruct the neuron, draw this convex hole, he can actually then start to look at the microglia within this convex hole and then profile the microglia that are specifically around D1 neurons or around the D2 neurons. And we've done this in stress, and what we've found when we do this microglia profiling, specifically, we actually found decreased density of microglia in our stress. This is our stress animals. These are the low social interactors, so these stress susceptible animals, compared to control animals that didn't undergo stress. We don't really see a difference between the high social interactors, so stress in general seems to you see a trend in reduction compared to control, so it seems to cause a reduction in microglia density around the D1 neurons, but no change around the D2 neurons. We also look at microglia branch points. So when microglia are phagocytic, they actually have less branching and they have bigger soma volumes. So what we found around the D1 neurons is there's actually reduced branching of these microglia, suggestive of phagocytic microglia around the D1 neurons in these stress susceptible low social interacting animals, but no change in the D2 MSNs. We've also looked at the soma. As I said, the soma size, larger soma, is indicative of more phagocytic microglia, and we do see this increase in these kind of stress-susceptible, low social interacting animals around the D1 neurons. Increased soma size of microglia around the D1 MSNs compared to these high social interacting animals, and a trend and increase compared to the controls. If you do a correlation analysis, we find that animals with with lower social interaction ratios have higher microglia soma volumes around D1 MSNs. No change due to MSNs. So it suggests that there's more phagocytic microglia surrounding these D1 neurons in these situations where you have these D1 neurons lose this dendritic complexity. I will say we haven't done this in the context of opioids yet or in any drugs, but that's something we want to start to look at. What's the role of microglia? What are they playing in these changes? Before I move on to our developmental stuff, I do want to mention, because the stuff I showed you before was bulk cell type analysis, but we have been shifting into these larger scale single cell analysis across many different conditions. This is just one example of opioids. It's actually in rats that self-administered heroin from David Dietz' lab at Kansas University. It's a collaboration with Seth Ament and his student Ben Grissom. Where we've put animals through heroin self-administration. These are rats, and then we've taken tissue either 24 hours after last self-administration or 10 or 14 days after last self-administration. This is actually not the nucleus accumbens. This is the ventral pallidum, and this has been taking a lot of time to do this study because, as you saw from Yoni's talk, the ventral pallidum is incredibly heterogeneous. So it was really hard for Ben just to catalog the cell types in the ventral pallidum. So we spent a lot of time cataloging them. You could see that you have this huge number of glutamate and GABA cells from this UMAP here, as well as other, and then one other smaller neuron population, this oxytocin AVP population, but then other types of cell types in the ventral pallidum. When you specifically look at the glutamate and GABAergic populations, you can see that there are many different glutamate and GABAergic populations, and they're actually segregated by enrichment of different neuropeptides in these different populations or different receptors. So this is just the basic characterization of all the cells. Now that we've actually cataloged those cells, we're now just starting to look to see what are the differences with this acute or prolonged abstinence from heroin. I'll also mention we have nucleus accumbens tissue that we've also done the same type of analysis, but that's part of a bigger analysis across species with many different groups, including Paul Kenny's group. So those, stay tuned for those studies. They're still ongoing, but I just kind of want to illustrate the type of work we're doing to understand these molecular underpinnings and how it can be challenging, especially in a place like the ventral pallidum that's so heterogeneous, to do these types of these cell subtypes analysis. So in the last bit of my talk, I want to talk about our work in the context of developmental drug exposure. This is through a substance use and pregnancy center that I lead along with my colleague Joe Cheer. This is a project on opioid use in pregnancy. We know in the U.S. that opioid use in pregnancy has increased, and the data is from 2010 to 2017, and it increased 130% during that time period. What happens with babies that were exposed to opioids in utero is you can often have preterm births, you can have low birth rates, stillbirth, birth defects, and you have neonatal opioid withdrawal syndrome NOWS. And it's thought that one baby is born every 24 minutes with NOWS. Later on in life, it's been difficult to dissect out what happens later on in life, but some studies have shown that there are neurodevelopmental delays, there's learning disabilities, there's emotional aggression, impulsivity behaviors, and there's also increased risk for mental health disorders. So we're really interested in these behaviors here, and really these long-term emotional outcomes from being exposed to opioids in utero. So to tackle this problem, we set up a fentanyl, perinatal fentanyl paradigm. This was actually established by Jason Alipio, and this is a collaboration with Asaf Keller, who's the chair of my department and colleague and a fellow Israeli. So when Jason was a student in Asaf's lab, he set up this paradigm where basically once a dam is pregnant, this is in mice, they're receiving fentanyl in their drinking water, or we have the water controls. The dams actually continue to get fentanyl until weaning because mouse gestation only covers the first 2 trimesters of the human gestation. So we wanted to cover that third trimester development of the mouse brain. So we had it in the mother's drinking water until P21. We also didn't want the moms undergoing withdrawal while they're nursing and taking care of their pups. And we've done a lot of work looking, after they're weaned at P21, we wait them to grow up to an adolescent age. We've done a lot of work looking at this adolescent stage. We have a lot of data in this original paper, but I'm just going to focus on the data on these more emotional behaviors. This was work done by Meg Fox in my lab, where she just did an elevated plus maze, and she found specifically in males that there is this reduced time in the open arms. So these males exposed to fentanyl throughout development, no difference in females, but we can appreciate there's quite a bit of variability there. With females, the one behavior we actually saw a difference in is the splash test. So you spray them with a sticky solution and you look at their time grooming. So that's thought to be like a motivation for self-care. And what we found is that's reduced in the females exposed to fentanyl. More recently, we're actually looking at these operant behaviors, sucrose consumption. We've taken animals during the adolescent period, and we've put them through this operant sucrose self-administration. First, start off with a FR1, where one nose poke, they'll get 1 pellet of sucrose, and then they have to work harder and give 3 nose pokes to get a pellet of sucrose. And we see that there is this blunting of this sucrose consumption in the fentanyl-exposed animals in these adolescent ages. And then if we put the animals back in the box 24 hours after this last sucrose self-administration, we actually find that they have this reduced seeking of that nose poke associated with sucrose. So that's thought to be reflective of an anhedonic response in these animals. We've then gone on in sequence, and this is bulk sequencing. We've looked at various regions across the reward circuitry, the prefrontal cortex, nucleus accumbens, VTA, but also a couple of sensory brain regions, because the SOF's been looking at S1 and ventral basal thalamus. And what you can appreciate is that this is actually showing differences in gene expression, or genes that are similarly changed between males and females. And what you can appreciate is in the nucleus accumbens, we see a lot of genes going the same directions in both sexes, but we see some going in the opposite direction. This is the prelimbic of the PFC. We see them pretty much going the same direction, as well as VTA and S1, but the ventral basal thalamus is really interesting, where they seem to be going in complete opposite directions when you compare males to females. You can also see this here, where you do this RRHO, where you can look at expression in these different quadrants. So if you get a signal in these two quadrants, you see concordant gene expression. If you get a signal in these two quadrants, that's discordant gene expression. And you can see in the prelimbic, the PFC, the VTA, and the ventral basal thalamus, the S1, we see this concordant gene expression between males and females, whereas the nucleus accumbens and ventral basal thalamus, we see this discordant expression. We've also done this big network analysis, and this is just showing different, what we call modules, where you see that genes that have high levels of correlations are, when you see significant modules, you see, you can identify genes that have high levels of correlations, but you're doing this analysis not with any biological input, but what you can do is go into these modules and identify biology. And what you can appreciate, so this is a Circos plot, basically showing, representing these different significant modules. And the outer circle is just the different modules that are color-coded. The first inner circle is the log fold change of males, and the log fold change of females, and then both sexes combined. And then these are the p-values. We can appreciate, sorry about that, is that in the nucleus accumbens, we're really only seeing male-specific modules, whereas in the VTA and prelimbic of the PFC, we're seeing both males and females or combined sexes. And as I said, we can go in these modules and find biological representations. We're seeing things like mitochondrial respiration, which is something we're really interested in popping up in the nucleus accumbens. Different developmental related molecules. So all this data, we're pursuing some of these molecules and some of these biologies. The other thing we've been doing, we're trying to get more cell-specific, so we're doing some spatial transcriptomics at P21 and P35, but we also want to do a temporal profile from P1 to P35. This is all bulk analysis using nanostring, so it's a targeted gene expression analysis. When you actually start to look at these different time points, we're not actually seeing them kind of factor out by fentanyl. We're actually seeing them factor out by their different developmental time points. And from this data, we can actually identify different clusters of gene patterns. And what we find, this is just showing the different clusters. We found 6 clusters, and these are just showing different profiles across time. And this is just the fentanyl water combined. But when we actually start to look into these clusters and look at the expression based on fentanyl exposure, in nucleus accumbens, we find more clusters early, or more significantly regulated genes in these clusters at these early time points. With the PFC, we're finding it kind of in the middle around P21, and the VTA, it's kind of all over the place. We're seeing these different developmental profiles depending on what brain region it's in. And then we're starting to look at the biological insight, biological ontology within these clusters. These are just nucleus accumbens ones. We're seeing things like mitochondrial respiratory chain complex, oxidative phosphorylation. And these are things we're pursuing. And again, we want to get more cell type specific. This is bulk seq, so that's for the future. So just to conclude, we've been able to look at opioid exposure across the lifespan, and we're always seeing these negative affective states, whether we have exposure early on or later on in life after this prolonged abstinence. We see this dendritic atrophy in these D1 neurons in adult exposure. We don't know what the structural adaptations are in the early life adversities. That's something we want to look at. We see these microglia changes in adult stress that are related to this D1 MSN atrophy, but we need to look at it in the context of opioids and in the adult and early life. And then we're seeing these cell type molecular signatures on adult opioid exposure that regulate dendritic complexity, but we need to profile these signatures further in the developmental time points, especially on a cell type level. But when but our first pass at looking at molecular changes in development, opioid exposure, we show that there are distinct brain region chains, temporal chains, and sex-specific profiles. So thank you. I think I finished just on time. I'm happy to take any questions.